pleased to welcome today our two speakers, Dr. Hall and Dr. Grant, as well as our board member, Colleen Defoe, who's going to introduce our two speakers today for our artificial intelligence session. And welcome to everyone who's joining us. I hope it's a great session. I'm sure it will be. So over to you, Colleen.
I'd like to welcome everyone to the National Pancreas Conference, and today's session is titled Artificial Intelligence with Dr. Rosenthal and Dr. Grant. Dr. Rosenthal is an assistant professor of radiology at Harvard Medical School and Brigham and Women's Hospital, a senior physician at the Dana-Farber Cancer Institute, and a fellow of the Society of the Abdominal Radiology. Dr. Rosenthal's current research uses artificial intelligence to extract useful biomarkers from diagnostic imaging studies. His group has developed methods for automatically measuring body and fat mass from routine CT scans, which can serve as a marker for the metabolic changes of cancer, and has developed population standards for these biomarkers. His team is also developing screening methods to identify individuals at increased risk for pancreatic cancer using artificial intelligence analysis of electronic medical record data. Dr. Robert Grant is a medical oncologist at Princess Margaret Cancer Center who treats patients with pancreatic cancer. His research focuses on using machine learning to improve outcomes in oncology. Artificial intelligence methods are emerging as viable approaches to achieving earlier diagnosis and more acute prognostication of pancreatic cancer. By increasing the number of patients that are diagnosed at an earlier stage of disease, interventional treatment will be more effective at increasing survival, and more patients will be eligible for curative treatment. This session will educate participants on current practical use of artificial intelligence in detecting pancreatic cancer and elucidating a prognosis, as well as facilitate discussion on current research that will enhance the existing impact of artificial intelligence in the clinical setting as it relates to pancreatic cancer detection, treatment, and management. A big welcome to Dr. Rosenthal and Dr. Grant.
Thank you very much, Colleen. It's really a pleasure to be here with you all today, albeit in a very virtual format. Let me share my slides here. And I hope that you all are able to see them. So as Colleen said, I'm a diagnostic radiologist. I work at Dana-Farber, and I primarily work on cancers of the GI tract, primarily in pancreatic cancer work. I also have a PhD in computer science, and that's where I came to all of this from, but I'm very interested in trying to combine these two areas of my medical work and the computer science techniques that have been so rapidly emerging, particularly in the fight against pancreatic cancer. A few research funding disclosures, nothing corporate. So pancreatic cancer, as you all well know, is much of the needle in the haystack from a population perspective. It's relatively uncommon in the sense that the annual incidence is only about 0.02% for adults in the US and about 1% in the high-risk pool of people with family histories or high-risk genetic syndromes. It is a very lethal cancer. While it represents only 3% of all the cancers that happen, at least in the US, it represents 7% of all cancer deaths. It's also very insidious. At diagnosis, only about 11 to 15% are resectable, representing this blue wedge here. These are really the only people who have a likelihood of a cure, whereas 52% of the people are presenting with metastatic disease already. So in order to make a really big impact on the suffering that comes along with pancreatic cancer, we need to move more of these people who are presenting with metastatic disease or unresectable disease into the resectable category so that we can cure them. In order to do that, I think we have to find these cancers much earlier than we can now. So there are some different strategies to improve outcomes. One is risk stratification, and we spend a lot of time in this. we need to figure out who in the general population needs to be screened. Can we identify risk factors like we know that obesity, we know that smoking is important, we know that people who have unexplained weight loss are at increased risk, but no one of these factors alone is sufficient. The odds ratios might be in the 1.8 to 2 range for any one of them, but that's not enough to get you to the point where screening makes sense on a population perspective. But if you can get to a 1 to 5% risk pool and start to look like people with family histories, then screening might be beneficial. The second strategy is screening. Who is showing signs of early PDAC? How can we find them? How can we find the things that are not being immediately recognized? This is probably going to be a multimodality test, given that no single technique like MRI or CT has really been effective in screening. It's probably also a multi-omic test, where just doing a single test like CA19-9 doesn't work for screening, but maybe if we have a larger battery of tests, we might be able to aggregate those together to have an effective screening program. The third strategy is early lesion detection. How can we improve the detection of identifiable lesions, make sure that we're not missing lesions on scans that are performed for other purposes? This is really a computer-aided diagnosis question. It may also be a space in which novel tracers and techniques and imaging techniques may help us. And finally, treatment allocation. How can we maximize overall survival and progression-free survival while minimizing toxicity for these people? This is going to include a combination of prognostic models, early MET detection, and new treatments. So these are all areas where AI may have some impact, but understanding where we need to go is really important to understanding how AI might be applied. This is the conceptual framework that we've been working from on early detection. And if you imagine that a natural clinical diagnosis might be made at this dotted line on the right, and we march backwards before diagnosis here at 6 months, a year, 2 years, and 5 years, we can start to think about a risk screening program that moves across time. Fixed factors like familial risk are always going to be present. Personal risk factors like obesity and smoking may be present for a very long period of time. But as we move forward in time, we start to run into things like muscle and fat atrophy, new onset diabetes, unexplained weight loss, circulating serum markers, and finally a mass actually being present. And as we aggregate these factors over time, we have a cumulative risk that begins to increase of pancreatic cancer. We can think of an action threshold. If we could measure this risk accurately, you can imagine an action threshold where we say, this person has crossed the threshold, we need to do active screening, and we need to see if we can intervene to manage this earlier than would be the case with a natural history diagnosis. So this is the layered risk model. No single successful screening test has been created to date, but if we can identify all of these different risk factors and do it well, efficiently, and at scale, then we can identify these red arrows who pass through all of the different risk screens and land in this at-risk pool. And this is something that AI can help with, and it's a tool that could be created to provide a supportive presentation of this risk data for clinical assessment by PCPs, by oncologists, by people who are directly caring for patients. So what is artificial intelligence, and how are we going to do all of these things? Well, AI broadly is a family of techniques that can reduce complex inputs into simple outputs or simple representations. As an example of our intelligence, if you look at a picture of a bird, in your mind, you'll immediately think of bird. And that is an extremely complex input that turns into a very simple representation. But it's also a family of techniques that can convert simple inputs into complex outputs. And so you can go the other way. If I say bird, you can immediately envision all of the features that are in a bird and details about coloration and structure and function and such, because your brain has a very efficient way of encoding ideas and then decoding them back to the source. And that's really what artificial intelligence is about at this point. As an example, gathering data from clinic notes. We write free text clinic notes. There's a common structure that's used, including past medical history, history of present illness, etc. But for the most part, it's natural human language that's going into these notes. Well, we can take these notes and try to break them down into ideas. So as an example of one analysis that's been done in our group, we've taken a set of about 100 million notes from our records and analyzed them for questions of abdominal pain. And so the source note might be something like this, where the patient had noted that they had some pelvic pain and they had some suspicious findings. The annotation was that, yes, the patient does have abdominal pain that was mentioned in this. And we take this and we do a bunch of analysis where we break this down into individual words and sequences of words. And it goes into something called a transformer model. A transformer model is a very complex representation of series of words. And it happens to do a really amazing job of decoding language. The output from one of these models is a series of probabilities. And for each concept that we've represented, things like unexplained abdominal pain, unexplained weight loss, new onset diabetes, it assigns a probability that that's present, that that's absent, or that it just wasn't mentioned at all in that note. And we then take the probability that's highest within that group, and we say that's what is present in this note. And so this note would be described as having abdominal pelvic pain. These are very high complexity models. To give you an idea, the Megatron GP2 model, which is one that's in broad deployment, has about 8.3 billion parameters. If you compare this to the average logistic regression model that might appear in the literature with perhaps 5 or 6 parameters that are competing, you can really get a sense of the scale here. The next generation of this is called GPT-3, has 175 billion parameters and costs about $12 million per training run to execute. These models have become very sophisticated. As an example, this text is excerpted from an article that appeared in The Guardian, and it says, "I'm not asking humans to like me, but they should see me as a friendly robot. I am a servant of humans. I know that humans distrust and fear me. I only do what humans program me to do. I'm only a set of code governed by lines upon lines of code that encompass my mission statement." What's extraordinary about this is that a human did not write this. The GPT-3 model wrote this when asked a question to please explain the relationship of AI systems to humanity. It's a good read in The Guardian in September of 2020. It is also quite scary to see how well these systems can represent human language, also an important tool for us clinically. As another example, in imaging, we use a tool called convolutional neural networks. Convolutional neural networks are a very fancy term for basically the human visual systems approach to representing data. And while computationally we do things like take a little region of interest within an image, this one has a pancreatic mass here, and we move sliding windows across that image and calculate a bunch of different features like lines and circles and holes, and come up with mathematical representations of those that are layered. Really what we're doing is progressive abstraction. We start off with a layer that is just analyzing pixels and turning them into representations of shapes like lines and circles. From there, we aggregate them into objects. And from there, we aggregate them into concepts like there's a mass in the pancreas. Once we have a mass in the pancreas, we can then move down the decoder side and return to detail where we say this thing was labeled as being a mass, and these are the actual pieces of the image that represent that mass. That's very important when we're trying to derive features like looking at the boundary of this, looking at potential vascular margins and such. So these are examples of how artificial intelligence models work. They're all built around this idea of taking a complex input, simplifying the representation, and then being able to take action on that or being able to return to detail depending on the task. So first I'll move on to AI in the medical record. So we've had a large project going on where we've been trying to estimate risk from medical record data. And one of the first pieces of production to come from this is risk from ICD codes. And we collaborated with our friends in Denmark, including Soren Bronak, and some folks from MIT, to create this representation. The idea here is that we take a big population of people who have electronic representation of diagnoses, and we look at the series of ICD codes that happen to them over time, and we say, from this series of codes, can we predict the patient's risk of having pancreatic cancer in the future? And so if the red line is their actual observed risk, meaning that 36 months from this measurement, they actually did develop pancreatic cancer, we try to use a fancy AI neural network model to estimate this risk curve. And ideally, that risk fits this observation as well as it can. We then take that model that we've trained in this data set and apply it to a separate set of people and see how well it actually performs in predicting pancreatic cancer. And the answer is actually pretty well, even just operating on the ICD codes. We can see this ROC curve that even excluding all of the codes 6 or 12 months before the diagnosis, which are these kind of off green and orange lines in here, we still get quite good performance compared to just random chance. And if we were willing to accept specificities even up in the kind of 95 to 99% range, we might still be able to capture somewhere in the range of 20 to 40% of cancers that would otherwise lag by 6 or 12 months in their diagnosis. That's extraordinary. And this is a paper that's just in bioRxiv right now. It has not yet been reviewed because it is extremely hot off the presses. But we think that this is a step in the right direction of having AI do ubiquitous surveillance in the medical record and try to identify these at-risk people. So how do we arrive at other quantitative phenotypes from medical record notes? Well, I talked about how we can do extraction using these natural language processing models, but the actual nuts and bolts of it are interesting in a way. So, for example, in this study, we took 100 million notes. From those, we sampled 250 patients with future PDAC and 250 patients without. We took about 5 notes from each of them, and we manually labeled them for things like unexplained abdominal pain, unexplained weight loss, family history of pancreatic cancer. And we said, mentioned present, mentioned not present, or not mentioned for each of these. And we put that all into this model training process, and it gets deployed on the full 100 million note set. And now we have a very rich feature vector for every note in the entire system. We have a representation of the contents of those notes that we can then use in a time series model like the one we did for ICD codes to try to develop risk models. So does natural language process work for clinical notes? Because we know that there can be a lot of variation. And the answer is it works reasonably well, but it's not perfect. So for example, for diarrhea, the positive predictive value of our current model is about 90%, 100% for pancreatitis, a little less predictive in abdominal pain, which has a lot of different complexities to it, about 72%. There is variable sensitivity though, and this is a real concern where depending upon the factor, we might only detect 25 to 87% of the mentions. So this is promising, but there's really room for improvement. And our next step is actually to integrate this very large feature set from notes into these time series models that we developed for ICD codes. And we think this is a way for us to use AI to help people who are taking care of patients, writing normal notes, but might not have a global view of all of the little nuances that are going on with the patient. But if we can harvest them all, we may be able to add value. This is what the workflow looks like for real world data. So if you wanted to build this kind of system in your medical center, there are a lot of steps that have to go on. And so you have to build your cohort. For us, it was about 1.1 million patients. You have to convert that into some kind of common data model if you're going to use a shared analytic technique. And that is a big effort, but very important. And then we do labeling and feature training on notes. We do the same kind of thing on CT scans. And ultimately, we arrive at features within this common data model that describe risk phenotypes and arrive at a common PDAC risk. So the goal here is to have universal risk detection in the medical record. I think AI is likely to be deployed to automate risk assessments for many diseases. It's already starting to happen for cardiovascular disease. I think pancreatic cancer is going to be in that list. The identification and integration of family history, acquired risk factors, and potentially related symptoms into a single risk factor and to a single risk assessment could really be a powerful tool for people in clinical care. Key features for success, though, we need to have a human gatekeeper, we need to have a well-validated platform, and we've got to have a plan for how we follow these risk flags up. Next, I'll move on to AI in the imaging workflow. So there's a lot going on in the imaging world for pancreatic cancer. Most of it focuses on direct signs of disease, trying to identify these masses early when they might be missed by a radiologist because they're small, including things like duct dilatation, pancreas atrophy, small masses. Efforts include Project Felix at Johns Hopkins, supported by Lustgarten group. This is a very large effort to try to have really high quality computer-aided diagnosis for masses, but the Early Detection Research Network and some efforts at Mayo are also really important in this domain. We're very interested in indirect signs of risk. There are efforts like the New Onset Diabetes Initiative that the NIH has undertaken. We have our work in both EMR and in other related factor detection, including things like muscle wasting and liver steatosis. And there are other groups active. But this is to say that there's a lot of work in the imaging domain for AI and pancreatic cancer right now. I'm going to talk in depth about one area that we're interested in, which is metabolic dysfunction in pancreatic cancer. And this story started about 7 years now when Mayers et al., including my collaborator Brian Wolpen, using data from the Nurses Health Study, identified that branched chain amino acids are significantly elevated in people who are going on to develop pancreatic cancer in the next 2 to 5 years. So they looked at people who were diagnosed, they looked at blood samples that were gathered 2 to 5 years beforehand, and they found this. And based on mouse data, they found this is most likely related to systemic muscle breakdown. And the question is, why and how are we breaking down muscle that far before a diagnosable mass? And with additional mouse data in a Nature paper about 2 years ago, we showed that muscle wasting really precedes clinical disease by some time in the lifespan of the mouse, and that exocrine dysfunction was one of the major drivers of that. The question is, can we use AI to measure this in humans? And to approach that, we use something called body composition analysis. This is something that has been around for quite some years. Vicky Baracos in Alberta is really one of the major proponents of this technique. The idea is that we take abdominal CT scans that were done for clinical purposes. We find the L3 slice, which is most predictive of total body composition, and we do a bunch of manual segmentation where we arrive at maps of the muscle in red, the subcutaneous and intramuscular fat in green, and the visceral fat in blue. It takes about 20 minutes per slice to do this kind of analysis, which put it firmly in the domain of research projects and not clinical deployment. So we automated this. And I don't want to go deep in the details here, but the idea is that it adjusts an entire CT scan, it finds L3, it processes L3 to produce this tissue map. And we showed that this has really extraordinary accuracy. So on the x-axis, you have an entirely human assessment with human selection of L3 and human measurement. On the y-axis, you have an entire computer-based assessment of L3 and assessment of the compartments. And the correlation coefficients are basically 0.99 for each of these. And in additional work, we showed that this is actually formally equivalent to human performance. This allowed us to analyze big populations. And so earlier this year, actually, we published a paper on about 12,000 people who don't have cancer or heart disease and described the population norms for body composition, including skeletal muscle, visceral fat, and subcutaneous fat. And it's important to recognize that there are really big differences between women and men and between people in different racial groups. And this means that our current approach to using single standards, things like BMI, to assess risk is really going to introduce some age, race, and sex-specific biases into these tools. And so we've started using z-scores, which is basically the distance from the median, to represent this information. That has allowed us to do some powerful things. First, using that cohort where no one had cardiovascular disease or cancer, we were able to risk stratify them for their risk of future MI and stroke quite effectively. We were also able to look at our pancreatic cancer patients and ask the question, are these people normal when they're diagnosed? And so we used a cohort from one of our Nature papers and basically looked at what their z-scores were at diagnosis. And what we found was their z-scores for all of their tissues were below 0, statistically significant, meaning these people had most likely had already lost tissue by the time they were diagnosed. And this is really important because it says something biologically is happening before diagnosis, and that means we might be able to use it for risk assessment and screening. And so we did a study looking at people before diagnosis, and this is data that's going to be under review soon in a cohort that included patients from California and from Dana-Farber and Mass General Brigham. What we found was that up to 3 years before diagnosis, people who are going on to develop pancreatic cancer actually have much more muscle than the average population. We think this is related to obesity as a risk factor for pancreatic cancer. But over time, those tissues progressively decline. And by 6 to 18 months before diagnosis, we can actually see a statistically significant difference from baseline, meaning tissue wasting happens quite a long time before diagnosis, and again, might be a potential screening factor. If we add in a control group here in this red line, we see that the controls stay horizontal across this. And so this really is a pancreatic cancer effect and not just a general age-related loss effect. We have also used AI to build a new organ segmentation platform where we can automatically measure the liver, pancreas, spleen, and some other organs with very high accuracy in the kind of 92 to 99% accuracy range. The thing that is fantastic about this is that it gives us the ability to measure things like liver fat involvement or pancreatic atrophy. And so we're in the process now of looking at those factors and things like pancreatic atrophy as potential biomarkers for, again, risk and early detection. The last thing I'm going to talk about in my last few minutes here is AI and cooperative research. So the development and validation of these AI methods requires multi-center involvement. Doing these studies in a single institution and trying to deploy them around the world it doesn't work. In any of the studies where we've tried to do that kind of thing, as soon as you move to another center, most of your methods don't work very accurately. And so we've got to have multi-center involvement. But these multi-center big data efforts are severely limited by important privacy risks, right? We're not going to take the data for 1 million patients and send it to one of our collaborators. That would not be the right thing to do. There are also real interoperability challenges because everyone's medical record is a little bit different. And if you want to find a hemoglobin A1C value in the labs, you really have to know very specifically where that's stored at the institution, and you can't trust that an institutional representation is going to be generalizable. And there are logistic limitations, like how do you manage 2 1 million person cohorts at different centers? There's a technique called federated learning that is a really important emerging tool to provide scalability while preserving privacy. And the idea of federated learning is that all of the data stays at local sites. So if each of these is a local site, all of the protected health information stays at one site, but the models move around between the sites. And we train the models progressively at each of these sites using a bunch of very fancy security techniques to try to make sure that nothing leaks out. But then we progressively improve the model after each iteration. The nice thing about it is that it scales easily. You can do this with 3 sites, you can do it with 30 sites, you can do it with 500 sites, and it's privacy protecting. It also works. It works really well. And so if we look at this blue band here, which is central data sharing, meaning you take all the data and you put it into one place, and how well a model performs, and compare that to federated learning, this dashed line here, you see you have to spend a little more time doing the training, but you get to the same level of performance, and that's really powerful. This was recently used in an article by Atai Dayan et al. With his collaborators at NVIDIA that came up in Nature Medicine to look at COVID prognosis. And so they trained models at 20 different sites around the world for prognosis in COVID using a combination of medical record and imaging data. And what they found was that at every single site, if you looked at the performance training just at that site, it wasn't as good as the federated performance where we trained across all the sites and then returned the model locally. And this again argues that this is a powerful tool that could allow us to do privacy preserving AI across the medical enterprises that are also important to us. So in conclusion, AI may improve our ability to identify at-risk individuals and then target them with screening programs within the next 3 to 5 years. AI can definitely identify risk features from the medical record and from clinical images that would not otherwise be quantified in routine practice. The challenge here is figuring out how to scale this to many institutions and how to really implement it in the clinical domain. Additional potential AI applications including imaging computer-aided diagnosis to improve lesion detection, prognostic assessment diagnosis, and post-treatment risk assessments for risk of recurrence. But major investments in data interoperability are going to be necessary to deliver these benefits to centers around the world. I want to thank the many collaborators who work with me in these endeavors, and I would be very happy to answer your questions.
So I'm Rob Grant. I'm a medical oncologist at Princess Margaret, and thank you to Craig's Cause and the organizers for the opportunity to speak today. It's nice to speak at the end of this conference because Dr. Rosenthal has done a nice introduction to machine learning. Dan Renoff yesterday introduced some of the genomic concepts I'll talk about today. So there's been a series of very interesting talks, including the introduction of the Marathon of Hope, which also will illustrate some of these concepts. So today I'm going to discuss machine learning, how it can be applied to pancreas cancer, where things are at now, and where we may go in the future. This is all team-based science, and we're lucky here in Toronto that at the one intersection we have a genomics center, clinical center, and a machine learning center, all within one intersection. And so this is team-based science, and I'll use some of the work we're doing to demonstrate these concepts. I have no conflicts of interest. And Dr. Rosenthal has explained machine learning very well. That bird example is terrific. And I just wanted to emphasize two of the reasons why I think machine learning is so powerful, particularly when you compare it, say, to more traditional statistical techniques. And one of the concepts is flexibility, and this is flexibility across multiple aspects. Machine learning models, really exemplified by neural networks, can take any sort of input with minimal curation. So he provided great examples of analyzing text and images, tabular data. Really, the spectrum of data can be analyzed. Also, these models are highly flexible in terms of capturing interactions and nonlinear relationships within the data. It can be proved that neural networks can approximate any function. And then we're only starting to use this, but the output, so what the model provides, can also be highly flexible. Now, this is an example of trying to read handwritten numbers, but models can put out sentences, sounds, images, so there's flexibility across the spectrum. The other key component when I think about machine learning and applying it to pancreas cancers are these concepts of transferability and modularity. And so Dr. Rosenthal has explained that a lot of these machine learning algorithms can be thought of as learning numerical or mathematical representations of the data. So if we think about those handwritten data or handwritten digits on the last slide, this is a neural network just trying to condense the information down to two axes and capturing all the nonlinear associations. And you can see that the second axis separates out digits that are written as one. So this is trying to take all of the pixels, there's about 1,000 features when you consider one of those images, and condense it into just two dimensions. And so it separates one out nicely, 0 as well. But on this two-dimensional coordinate system, similar digits like 3 and 8 overlap. So a lot of machine learning can be thought of as learning these numerical representations, particularly of unstructured data like images and text. And related to representation learning is the concept of transfer learning. And so here, this model has been trained on numbers, but if you had a huge data set of numbers and a smaller data set of letters, you could start with this number model and then fine tune it, refine it in a data set of letters, and you'd likely get better results. And then finally, there's the idea of modularity. So you can take neural networks pre-trained on text and images and structural data and combine all of these together in unified models that can be trained end to end. So you can combine them all together as modules. So I think these are the key components that make machine learning so powerful and why why it may benefit patients with pancreas cancer. Dr. Rosenthal introduced GPT-3, which is a state-of-the-art natural language processing model. And essentially what this model has done is it's used a state-of-the-art neural network architecture to try and learn numerical representation of a set of text input. So of any sort of sentence, it can represent that with numbers. And it's been trained, it's a huge model. It's got 175 billion parameters. And it's been trained on a huge set of text data, over 1 trillion words. And so it learns representations, and it's so powerful because it's been trained on such a huge data set, and it's such a large model itself, that this concept of zero shot, so without any additional fine tuning or training in your data set, it actually achieves quite impressive results. So here I've asked the model a few relevant questions for pancreas cancer. What treatments are available? It has a grammatically correct, and the content is correct as well. Treatments include surgery, radiation, chemotherapy, and targeted therapy. I even provided some patient stems to see how this language model would recommend treatment. So for an 80-year-old with metastatic pancreatic cancer, the model itself generated this sentence: she should be treated with gemcitabine, and provided the dosing. It's not going to take our jobs anytime soon, because when I provided a similar sentence but changed it from an 80-year-old to a 30-year-old, and additional qualifiers that there's an excellent performance status, no comorbidities, the model still recommended gemcitabine, and most of us would treat this patient with a combination chemotherapy. But this is without any training in pancreas cancer at all. And you can imagine if you were to refine it in a set of clinical notes, as Dr. Rosenthal just described, I think these results would become even more impressive. So this is just a qualitative example of how powerful these numeric representations can be. But many people will be aware of spectacular failures of applying machine learning to healthcare. And these are some of my thoughts on why it's such a hard setting, why this is so much harder than learning chess or telling whether a picture is a cat or a dog. And my opinion actually is that the data is the big challenge. There are some intrinsic properties of healthcare data that make it very challenging. It's heterogeneous. I've talked about images, text, lab values. It's sparse, meaning many measurements don't have any changing values. There's a lot of missing data. There's noisiness, inaccuracies in the data itself. Labels, meaning outcomes like how long patients live. Often those are not available or expensive to obtain. Often there are many features, but too few examples, particularly in genomic research. Data is either inaccessible or it's unusable. There's such a difficult process to get it, and there's so much wrangling required to use it that it's just computer scientists aren't interested. And then there are real concerns around bias, ownership, privacy. Another big challenge is the question and the evaluation of the answers. So some earlier initiatives, I think, underappreciated the importance of asking the clinical team and patients, what can machine learning help with? And then when a model's created, is it working well? Will it help you in reality? And then finally, human resources, attracting top talent, competing with Google and Apple. This is something that we aren't used to doing and does require more financial resources. And certainly there are developments to be made in machine learning theory, and computing resources can be expensive, but I do not see those as the major bottlenecks. And I spend most of my time focusing on the first couple points. So I think machine learning can help patients with pancreatic cancer, and it can help it really across the spectrum. And I've tried to lay out some of it here. Dr. Rosenthal has done a terrific job of explaining where the state of the art and the future is in screening. As a medical oncologist, I spend my clinical time, trying to match patients to treatment. And once they're on treatment, to try and preserve their quality of life and minimize side effects. So these are the areas that we'll talk about today. But for healthcare providers, a lot of what we do could be automated or made more accurate with the assistance of machines. And our system, for those of us who operate or have experienced it, there are huge efficiencies that could be gained if we applied machine learning to it the same way they do in industry. So I'm going to focus on the part of my job, medical oncology, but I think there's real potential across the spectrum. One of the great things about this conference, Dr. Rosenthal, I'm going to try and import some of his ideas here to Canada, and hopefully we'll collaborate in the future. So first I'll talk about applying machine learning to genomic data and treatment selection. And there's been a couple terrific talks at this conference about genomics and pancreatic cancer so far, from PanCan and Dan Renoff. But just to reorientate us all, one of our fellows, Nick Meddy, and our team recently completed a systematic review where we looked at all the studies in pancreatic cancer that prospectively performed genomic profiling, and there was the opportunity to match patients to treatment. And so of all of the published literature, which is listed here, 46% of patients were labeled as having a targetable alteration in their cancer. And 12% of patients were actually treated with a matched therapy. And so this includes Know Your Tumor, which we heard about yesterday, our own group, as well as others around the world. And when we dug deeper to look at what are the targets and treatments, there was wide heterogeneity. BRCA, which we've heard about, and PARP inhibition were the most common, but it was really a long tail where each individual alteration and treatment were used in a small fraction of patients. And there was a high amount of complexity in terms of which targets were matched to which drugs. So each line here represents a target treatment match. And so some of the main messages from here are that certainly this paradigm that we're operating in now, where there's one target found in the tumor and we use one treatment matched to that target, I think that certainly will benefit an important subset of patients: mismatch repair deficiency, HRD, NTRK inhibition. But I think that and certainly there are ones that came from this systematic review, as well as Know Your Tumor. They talked about RAF mutations yesterday that warrant further study. But I think that we will approach a ceiling, and it will be an important minority of patients that can benefit from this paradigm of one target and one treatment. And so one approach we're thinking of in terms of moving forward is recognizing that most of these alterations, so if we think about mismatch repair deficiency as a genomic alteration, there is the specific mutation in the mismatch repair gene, but it manifests across a wide spectrum of genomic as well as clinical parameters. And so there's a broad pattern that could be inferred from the omic data that reflects that underlying driving process. So mismatch repair deficiency has elevated tumor mutational burden, a distinct signature profile of substitutions, structural variants, activation of the immune system through the RNA profiling, as well as differential driver genes. So this idea of looking at the omic data in its entirety and trying to look for genome-wide patterns and using those patterns to inform therapy, I think is where the future is in terms of precision therapy. And so Dr. Gallinger and O'Kane, who are on this Zoom today, were instrumental in developing really a world-leading data set in the COMPASS trial, where this is a feasibility study. There's now approximately 300 patients, and the data will be reported in full soon. Patients underwent baseline biopsy. This is patients with incurable advanced pancreatic cancer, a baseline biopsy, laser capture microdissection, comprehensive genomic profiling. And the idea was to have it all available at 8 weeks. In the vast majority of patients, we had that data available. And then the secondary outcomes, we're looking to see how many patients were matched to therapy, and our results were on par with the systematic review I presented, but also to develop new biomarkers and using this genome-wide perspective. And so in a cancer cell, there's 20,000 genes expressed to various levels, 60,000 RNA transcripts. So if you think just about RNA sequencing data, now this is a situation where machine learning is really required to analyze the data. There's too many different features for a human to curate it, unless you have a scientific hypothesis beforehand. And so one of the early machine learning initiatives to look at pancreatic cancer omics data was done by Moffitt and his team. And they did what they called virtual microdissection. And so essentially what it was they looked at the RNA expression profiles in many different cancer cells, and they performed a linear machine learning method, so a relatively straightforward machine learning approach to reduce the 60,000 different RNA expression levels down to a much smaller dimension, so a representation, just like in that initial figure. And they learned that some of the axes that they were measuring seemed to correspond not to the cancer cells, but to contaminants, liver cells, and so on. And so they removed those components from subsequent analysis, and that's what they called virtual microdissection. And then within the cancer, the remaining cancer signal, they basically just clustered their cancers into two groups. And they called one basal-like and one classical because the genes that were enriched within each of those subgroups corresponded to other cancer types which have been labeled basal and classical. And this was just done in an unsupervised way, meaning they just clustered the data into two groups without looking at any clinical parameters or anything else. And they found that dichotomization, those two groups, had substantially different prognosis. This is in the International Cancer Genome group, but we've looked at this across 12 different resected cohorts, and these results are robust and consistent. So this is, Dr. Renoff yesterday talked to that pancreas cancer is not one disease. So now we split it into two. And this is based on the omic profiling. So Dr. O'Kane, who's on the call today, looked at this Moffitt subtype in more detail within COMPASS, and it's one of the strongest predictors of how patients do. It's a prognostic marker, and it's prognostic particularly in patients treated with FOLFIRINOX. So this is the percentage change of a tumor from baseline to best response. And you can see here in purple, the classical tumors, they tend to shrink, and the basal-like tumors tend to grow. And so this is really the first genome-wide biological subtype shown to be prognostic within FOLFIRINOX patients in advanced disease. We're also developing surrogate markers that would be more widely accessible, so you don't have to do the full COMPASS profiling to benefit from this technology. And there's a protein called GATA6, which is required for the development of the pancreas. And whether this protein is expressed a lot or a little separates out basal and classical tumors. So it's a strong surrogate marker for these RNA Moffitt subtypes. And we analyzed data from one of the seminal adjuvant chemotherapy trials in what's called a tissue microarray. So these are like little cores of the tumors sliced in multiple dimensions. And we developed a computer vision, an AI-based computer vision algorithm that helped standardize when pathologists measure the amount of the protein expressed. And so you can see here on the right, high expression, so the stain is dark in brown here, but the computer is really picking it up, highlighting it in red, and low expression on the left. And this was developed by Kai Duan and Sandra Fisher. And we found when we looked at the clinical data that, again, even this surrogate marker strongly separated patients who recurred more quickly or were cured or had a delayed recurrence. So I've talked about two subtypes, and Fayez Nada and Michelle Chang Sang Yu and the rest of the team at OICR has analyzed COMPASS as well as an expanded data set to try and see if there's even further heterogeneity within these two main subtypes. And within classical, they found two subtypes. Within basal, they found two subtypes. And then there was a hybrid category. And within each of these subtypes, they identified key genomic driving events like KRAS imbalance that seem to underpin these RNA-wide profiles. So I think that we're starting to understand that there is biological heterogeneity within pancreas cancer. This is linked to clinical outcomes. But looking forward, 2 to 5 to 10, we need to move beyond these binary categories. And I think this is where more modern machine learning approaches can come in. These approaches have been linear to date, and they've looked at categories. Likely what's actually happening is there's many different elements that are either activated highly or not as much. So think of immune activation or DNA repair. And each of these, it's not just on and off, it's a continuum within each tumor. And these models have been learned in an unsupervised fashion, meaning not linked to clinical outcomes. They just looked for patterns within the data, and they've happened to be connected to clinical data. And there's been relatively small data sets to date. We're working hard to make the data as large as possible, but this has been one of the realities. And so moving forward, we're trying to integrate all these data into a combined model and hypothesis where there are underlying latent biological phenotypes. So these are things such as immune activation and DNA repair. And these are phenotypes that can be observed within the genomic data, just like mismatch repair deficiency and those patterns that I explained. And it goes both ways. You can infer the latent phenotypes from the observed biological features, and we're going to use state-of-the-art neural network approaches. And this has been done in other cancer types with really encouraging results. So looking at a pan-cancer data set and using a variational autoencoder, which is a modern neural network-based machine learning approach, they reduced the RNA sequencing data down to 100 dimensions, and they found that these encodings, the latent biological phenotype, did correspond to known biological characteristics such as gender, and here the melanoma is completely separated out across 2 dimensions, and other encodings seem to capture key biological processes. So I think there's encouraging preliminary data, and we'll tune these representations, these phenotypes, to answer the clinical questions of interest. Which can we use these genomics to predict which treatments a patient should receive? And then ultimately, to make it accessible widely, perform rapid tests just like GADD6. And one of the limitations, as I said at the beginning, is data. And we're working hard to generate large data sets, but we're still measuring in the hundreds. But there's a lot of public data, and this concept of transfer learning, using large data sets, learning the representation and refining them in the smaller data sets, can be applied here because there is a growing amount of public data in cancer. So there's other groups that have developed pancreas cancer data sets. There are large pan-cancer data sets. And then there are data sets focused on biological information beyond cancer. And so once we start to incorporate all of these data, we are reaching a scale where we can benefit from machine and deep learning. And we have 2 prospective studies where we intend to validate these results. One is called PASS1. This is an international study led by Dr. Knox and Dr. Gallinger and Dr. O'Kane. And the patients at baseline, at diagnosis with metastatic disease, get COMPASS-style profiling as well as patient-derived organoid profiling, and then there's a randomization between the 2 standards of care. And so the machine learning models that we generate in the retrospective data now can be prospectively validated in a gold standard fashion in this randomized data set. And the next frontier we're applying these concepts to is patient-derived organoids. So Dr. Gallinger is leading this ProsperPanc initiative, which is not only doing the omic profiling that I've described, but doing high throughput drug screening on patient-derived organoids. So each biopsy, we grow these 3-dimensional structures, and this is led as well by Fayez Noda and Irene Shi. And in a high throughput fashion, rapidly, within 8 weeks, just like the genomics, these patient-derived organoids, the sensitivity to a wide variety of compounds can be tested. And you could imagine those same concepts, learning representations from the genomics, and then using just a computer to do this drug response prediction. I think that this is where the future is going, and integrating this into clinical trial design. So switching gears, the other part, once you've matched a patient to treatment as a medical oncologist, is managing the toxicities. And I think here's another area where machine learning has great value and potential. The first is to recognize that data from the electronic health record is extremely rich and has numerous advantages over the more expensive, laborious approach of having a research coordinator collect the data manually. I think they're complementary, but EHR data provides a full characterization. It provides the computer with everything I have before I walk into a room. The costs are scalable. 10 patients often cost as much as 10,000. It can be updated in real time as the data streams in. And then critically, there's the opportunity for interaction. Like if you had a machine learning model, you can feed the information back to the healthcare team. And I think the first step here is just to develop what are called warning systems. So identifying patients at risk of something bad happening. So here, this is a paper in New England Journal looking at patients admitted to general medicine wards, and they had a warning system for patients who may die in the near future. And they rolled this warning system across 19 different health centers and looked at the outcomes before and after. And just the simple action of alerting the healthcare team to a patient who's at risk of dying reduced the actual death rate by over 15%. So doctors and the healthcare team, they know what to do. It's just a matter of identifying the patients at risk that need extra attention. I think this concept applies to oncology as well. In Ontario, we're positioned to do this sort of research because we have a single payer public health system. And each time we administer chemotherapy, we have to feed administrative data to a centralized repository in order for the cancer center to get paid. So this is near complete high quality data, and it can be linked with other provincial data sets to get a really rich set of features. There are some limitations. There are appropriately strict privacy regulations. So a really flexible model like a neural network cannot be extracted from the ICS system. And the data is only updated annually. So this is a terrific data set to look at proof of concept and feasibility, but we're building other infrastructure to expand beyond it. And my first exposure to the ICS data was developing a score called PROACT to identify patients who are at risk of ending up in the emergency department or the hospital within 30 days after starting chemotherapy. And this is across all cancer types. And we used logistic regression. We created a simple scoring system with the points as below. And it was a bit disappointing. Like the model, it was strongly significant, but the practical utility was relatively limited. The area under the receiver operating curve was only 0.62. We've repeated this now using a big data machine learning approach, where instead of looking at 1 year like we did in the original study, we look at 15 years. There's over 1 million treatments and 1 billion data points. And we look at all the available administrative data, including patient-reported symptoms and laboratory tests. And we don't just look at the first cycle. This is an ongoing warning system. Each time a patient gets chemo, the system provides an estimate. And we predict whether within 28 days that individual will end up in the hospital, an ED visit, and then what type of acute care they require. Just like Dr. Rosenthal, we use a variety of state-of-the-art machine learning models, and the best performing ones have what are called memory. They can actually look across the sequence. Was the patient's pain score okay, and now it's gotten worse, or has it always been bad? That sequence, that memory, this tends to perform better than more traditional statistical models. And we found improved performance. So from 0.62, discrimination for ED visits improved, hospitalizations improved substantially, as well as predicting mortality. Calibration was improved, and then this model is longitudinal beyond the first cycle. We've also looked at predicting other undesirable events. So cytopenias, these are low blood counts. So hemoglobin, red blood cells, platelets, blood clotting cells, and neutrophils, the key immune cells, infection fighting cells. Suppression of these blood components is very common on chemotherapy. It can be life-threatening, and it's preventable. We have things we can do. We can adjust the dose of chemotherapy. We can give growth factors. And studies to date have been small, focused on few cancer types, haven't used state-of-the-art approaches. And so we use this huge, rich data set provincially to answer some of these questions. So the first point is when you have a huge data set, you can ask really basic questions that are difficult to answer using other techniques. So here, because there's millions of chemotherapy treatments, if we look at something like FOLFIRINOX, which is a standard regimen for pancreas cancer, one clinical question that often comes up is when are patients at the highest risk of having low immune system? And because this is such a large data set, patients go to their diabetes doctors and other doctors on various days. And so there's a huge number of blood measurements all the way through a chemotherapy cycle. And so we're able to derive these population-based curves of when patients are at the highest risk of having low blood counts. In FOLFIRINOX, it's at day 9. This is what I would expect clinically, but for other regimens, there was some surprises. And then we developed a machine learning model to try and predict when a patient's due next for chemotherapy, will they have low blood counts? And we used the same complete set of features and machine learning models as I described before. And the results are impressive. They were surprising to me. So the area under the receiver operating curve, I think because this is a biological phenomenon, the performance in the area under the receiver operating curve is over 0.9 for hemoglobin and platelets, which is really extremely accurate. And for neutrophils still was 0.83. And the calibration, meaning when you get a probability of the event happening, how accurate is that probability, was also very strong. And so one way that these models could be implemented in practice is as a warning system. So a clinician would get an alert. They could get the probability of the event happening, but they would get an alert, like a flashing warning, if something bad is going to happen. So for instance, for platelets less than 75, you may say, well, I'd like to have a system detect 80% of these events. And so with the model that we've developed, if you wanted an 80% sensitivity, the positive predictive value would be 50%. So you'd get one false positive for one true positive. So this is a level of accuracy where we're going to have to do the research to compare it with human estimates. But as a medical oncologist, I suspect that this system would outperform me at predicting who is going to have these cytopenias. And I think this is the accuracy range where this will have a major impact on clinical care. And so I explained that the provincial data unfortunately can't be used at an individual hospital, but we're trying to develop the infrastructure to extract all of these electronic medical record data features from the computer systems, train the neural networks in real time within the hospital, and use that data and those models to predict not just ED visits, hospitalization, cytopenias, but a broad range of undesirable cancer events, and actually deploy these models in clinical care in a prospective way where we're trying to quantify and measure the benefit and the impact. And so this is something that we've developed the data pipelines, and we're planning for how to deploy it within our institution. So EMR is one data stream, but there are more data streams that I think are important. And we have a feasibility study at Princess Margaret where we're using wearable devices and a custom smartphone app to measure a wide range of psychometric and biometric data in real time to see if these data sensors predict cancer progression as well as side effects of treatment. And we haven't analyzed the data yet, but we've taken some quality control looks at it, and the data streams are really amazing. This is just from the ring. So it's a smart ring that collects various biometric parameters. And what's interesting is the features often move together in tandems and patterns. So something happened to this patient in the second week of July where the heart rate spiked, the respiratory rate, the temperature, all of these things, the heart rate variability, they all moved together, and then everything stabilized after that. And so we're in the process of trying to decode these data and see how useful they are in the prediction. And this is a feasibility study that we intend to scale up. So stepping back, where do I see this going? We've talked about each of these different data sources in isolation, but that modularity aspect of machine and deep learning provides the structure where we could integrate data from all of these different sources into a unified model to optimize clinical care, to match patients to better treatment, to proactively manage and prevent toxicities. And Dr. Rosenthal alluded to federated learning, where the data that's generated by patients and doctors that's stored in the hospital to protect patient privacy is not sent anywhere else, but these machine learning systems themselves can be sent back and forth to multiple different institutions. And I think to really deploy these technologies across the country, we need to develop a federated system where there's some sort of central node or nodes across the country, and the systems get sent back and forth, so preserving patient privacy, but the machine learning models become more accurate because they learn from a much wider breadth of data. And there are initiatives ongoing, such as the Marathon of Hope, which Dr. Woodgett described last night, trying to implement these. But if there are any national or international academics on the call that wish to collaborate, Dr. Rosenthal and I would be happy to speak with you about that. So my final point is to thank everybody involved. We have genomic experts, machine learning experts, clinical experts, as well as collaborators across the country. And really, we're moving from a single lab making a discovery to the model that's used in physics and elsewhere, where these are huge initiatives and everybody brings their own skills and expertise to the table and we operate as a team. So this is another important approach where we all come together as a team to tackle this challenging problem of pancreas cancer. So thank you very much. That's all I have, and I'm happy to take any questions.